Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 7.5, Hardy-Weinberg equilibrium. So today is pretty simple. We'll be talking about the conditions for Hardy-Weinberg equilibrium as well as the actual Hardy-Weinberg equation. And our suggested skills are going to be concept explanation, 1C explain biological concepts and processes in applied contexts, and statistical tests and data analysis, 5A perform mathematical calculations, including mathematical equations in the curriculum, such as Hardy-Weinberg equation, means, rates, ratios, percentages, and percent changes. So Hardy-Weinberg equilibrium, invented by Hardy and Weinberg, represents a non-evolving population for a particular locus. So that means that for that particular location on the chromosome, for that particular gene, there is not going to be change in the population over time. And so the allele frequency for that gene is going to remain constant. And so the null hypothesis here is that there's no significant difference between two populations. And if one is in Hardy-Weinberg equilibrium and one isn't, that means that the other population is evolving, and our null hypothesis has been rejected. But in order to achieve Hardy-Weinberg equilibrium, there have to be these 5 conditions that are met. And these are the 5 causes of evolution. There can't be mutations because mutations will add random genes that can then be selected for. There's no sexual selection. Mating has to be random. No natural selection. No specific traits are selected for in the environment. No genetic drift, meaning that the population size has to be large in order to protect against genetic drift that could reduce the genetic variation within the population, and no migration so that there is no gene flow. So it's obviously impossible to achieve these conditions because evolution is always occurring and environments are always changing, as well as some species are just set for sexual selection, and not all populations are large and migration does occur, but this is kind of representative of the ideal state in which there is no evolution occurring, and this is Hardy-Weinberg equilibrium. And so we have two parts to the Hardy-Weinberg equation. So firstly, you need to know that P is considered the frequency of allele 1, usually used for whatever the dominant allele is, and Q is the letter that represents the recessive allele, the frequency of the recessive allele. And when the gene has 2 alleles, the frequencies add up to 1. So within the population, there can only be enough alleles that make up the entire population. So the allele frequencies of P and Q, the frequencies of the dominant and recessive allele, make up 1 because there are no other alleles within the population. And the frequencies of how many homozygous dominant individuals, how many heterozygous individuals, and how many homozygous recessive individuals, so now we're looking at different genotypes, also add up to 1. And that's what's shown by the other part of Hardy-Weinberg equation, p squared, so for a homozygous dominant, meaning 2 dominant alleles, plus 2pq, that's for our heterozygous individuals, plus q squared, that is going to be for the homozygous recessive individuals. Those frequencies are also going to equal up to 1. And so questions are often going to pair Hardy-Weinberg equations with the chi-square, asking are the expected population results under Hardy-Weinberg, meaning so are the populations that are not evolving different from the observed. So essentially, is the difference between the two populations enough to show that evolution has occurred? And if you need some more review on chi-square, you can take a look at our video for 5.3. But for now, let us practice some Hardy-Weinberg. So now we're going to look at a population which is under Hardy-Weinberg equilibrium, meaning it is not evolving, the allele frequencies are not changing, it's remaining constant. And so you've sampled a population in which you know that the percentage of the homozygous recessive genotype, lowercase a, is 36%. And using this information, we need to calculate the following. Firstly, the frequency of the homozygous recessive genotype. Well, it was just given to us in the problem. That is 36% or 0.36. Now, when we're asking for the frequency of the recessive allele, we have to look at our Hardy-Weinberg equations. We know that our frequency of the AA genotype, that 36%, is represented in the Hardy-Weinberg equation by q squared, like we discussed on the previous slide. So if q squared is equal to 0.36, that means that Q by itself is equal to 0.6. Because 0.6 times 0.6 equals 0.36. So to get rid of the square, we square root both sides. And so with Q being 0.6, that shows us the frequency of the recessive allele. And now we want to know also what the frequency of the dominant allele in the population is. And we know that the recessive plus the dominant alleles in the populations have to equal 1. P plus Q have to equal 1 in the population. And we know Q now, we know that Q is 0.6, so P plus 0.6 equals 1. We subtract 0.6 from both sides, we get P is 0.4. And so that's going to be the frequency of our dominant allele. Lastly, we need to find the frequencies of these other genotypes of AA, so homozygous dominant, and also of heterozygous. And again, we can turn to our Hardy-Weinberg equation. Starting off with homozygous dominant, in the Hardy-Weinberg equation, that's represented by P squared. So if P is equal to 0.4, as we just determined finding the frequency of the dominant allele, then P squared is going to be 0.4 times 0.4, or 0.16. So our frequency of the genotype of the homozygous dominant genotype is 0.16 or 16%. Lastly, we need to find the frequency of the heterozygous genotype, and that is in our equation as 2pq. 2pq. And we know both p and q now. We found the frequency of both the dominant and recessive allele. So 2 times p, which is 0.4, times q, which is going to be 0.6, results in the answer of 0.48. So our frequency of our heterozygous individual is 0.48, or 48%. And now, in order to check our work, we know that all three of these frequencies have to add up to 1. So our 36% plus our 16% plus our 48% has to add up to a total of 100% of the population. And doing this math, 6 plus 6 is 12, plus 8 is 20. 2 plus 3 is 5, 6, 10. So 100. We have calculated the 100% of the population and each of the frequencies of the genotypes and of the alleles, which also are going to be adding up to 1. 0.6 plus 0.4 makes a total of 1. So the allele frequencies add up to 1. The genotype frequencies add up to 1. That means that we have successfully used Hardy-Weinberg equation to calculate the frequencies of these genotypes and alleles in this non-evolving population. So, we need to know conditions for Hardy-Weinberg. Essentially, no evolution is happening, no genetic drift, no gene flow, no sexual selection, no natural selection, and no mutations. And Hardy-Weinberg equation, P plus Q, so our dominant plus recessive allele frequency is equal to 1, and P squared plus 2PQ plus Q squared, our genotype frequencies also equal up to 1. This was Hardy-Weinberg. We'll see you in 7.6.